Hello everyone, and welcome to the first in a series of biology review videos that are meant to help students review essential content for the end of course SOL biology test done in Virginia. But these can be used as a refresher on a lot of basic biology topics for exam review if you're coming from another state. As you follow along, I encourage you to use resources that are linked in the video description, and go ahead and subscribe if you find this kind of material useful for biology science exam review. First up in the Virginia course description for biology, the very first content topics cover biochemistry and life processes. So in this section, we're going to talk about water, macromolecules, enzymes, protein synthesis, photosynthesis, and respiration. And then after that, the next section is going to cover cell structure and function. So there we're going to talk about cell theory, organelles, cell cycle, cell transport, and cell specialization. So water, remember, is really important to living things. It's got special chemical and physical properties that basically allow life on Earth to exist. It is the universal solvent, meaning it dissolves lots of different chemicals and minerals and nutrients that support life. Water is a polar molecule, meaning it has a property that allows them to be strongly attracted to each other, which also helps us with a high surface tension, meaning it doesn't break easily, and cohesion, meaning it can stick to itself. Water also has a high specific heat, meaning that it takes more energy to increase the temperature of water compared to other substances, which helps us regulate our temperature in all living things. Water makes up 70 to 95% of organisms, and it consists of 1 oxygen and 2 hydrogen atoms per water molecule. So there are some processes that all living things go through, including growth and repair, reproduction, gas exchange, metabolism, and response. And there's going to be certain macromolecules or certain larger molecules that are going to play a role in all of these processes. And in those biological macromolecules, carbon is a key ingredient. Now, actually, carbon, hydrogen, and oxygen are going to be present in all our main categories of biological macromolecules. Our 4 main categories of organic compounds are nucleic acids, which are made of nucleotides, proteins, which are made of amino acids, carbohydrates, which are made of monosaccharides, and lipids, which are made of usually glycerol and fatty acids. Now remember that all 4 categories of these biological macromolecules contain carbon, but we're going to talk specifically right now about proteins, which is one of those categories. And within the category of proteins, we can find enzymes. So enzymes are special types of proteins that are going to lower the activation energy or reduce the energy it takes to get important reactions to occur in living systems, in living things. So some important things to remember about enzymes is that they are proteins. Enzymes are specific to the molecules that they react with. After reactions, they remain unchanged, so they're reusable, and they work in specific ideal conditions or a specific environment. So for example, if the pH is too high or the temperature is too high, these enzymes could mess up and not do the jobs that they're supposed to do. A key word you can remember for enzymes is a catalyst. Think of it as something that gets reactions going. And there's lots of different enzymes throughout all of our cells in our body that are going to help us do lots of different important chemical reactions within the body. Now remember, another one of those important life functions is metabolism, or how we get the energy we need from other things. So in organisms with mitochondria, there's a process called cellular respiration that's going to help us take glucose and oxygen and use that to make ATP or energy for the cell. As a byproduct, carbon dioxide is also produced, and then of course there's water that comes out of that reaction as well. Think of it on a larger scale. When we breathe in air, we're taking in oxygen that's going towards that process of cellular respiration. And when we consume foods or when we eat, that glucose we take in is going to cellular respiration as well. And when we exhale carbon dioxide, that's coming from the process of cellular respiration. And the energy we need to move our bodies and keep our cells healthy, that's the ATP that we use in cellular respiration. And then, of course, water. We can see water vapor as we breathe out. That is from cellular respiration as well. Now, if we put the reaction like this straight across, we can remember glucose and oxygen yield or produce water, carbon dioxide, and ATP for energy, which is kind of the flip or the reverse of another important energetic reaction, photosynthesis, which organisms that have chloroplasts perform. So things like plants and green algae, they're going to take in carbon dioxide and get sunlight energy and water to produce glucose and oxygen. Now it's important to remember that plants perform cellular respiration too, so they're going to use some of that glucose and oxygen themselves, but what they don't use it gets combined to form larger molecules that we can eat later on. And then, of course, the oxygen, plants produce an excess of oxygen, so they release it into the atmosphere and we get to breathe it in and use it in cellular respiration ourselves. So these two processes are the reverse of each other, but they also sort of feed each other. All right, let's go back to some of these major macromolecules. We're jumping back and forth a little bit to connect our ideas, but nucleic acids are another really important category of organic macromolecules or biomolecules that living things need. In fact, all living things contain nucleic acids because they all contain DNA. So DNA is found inside the nucleus of eukaryotic organisms, or in what's called the nucleoid, which is kind of like a mass of DNA in prokaryotic organisms. The structure of DNA is made of nucleotides, and each different nucleotide, whether it's A, T, G, or C, is going to pair with another type of nucleotide. So it's important to remember that A always pairs with T and G always pairs with C. Some people remember this as apple tree, good cookie, or all teachers can go if you want to say it backwards. But whatever you need to remember, or I like to think of the letters with straight lines pair up and the letters with curves pair up. Here's a close-up of a nucleotide. Remember, it's made of one of those bases, A, T, G, or C, a sugar and a phosphate. And together, those sugar and phosphates, here as S and P, are going to form the backbone of DNA. And we put it all together. This is what our double helix starts to look like. So one side of DNA is here, and then again, it's going to pair with another nucleotide on the other side to form this structure with two backbones, and then the base is paired in the middle. And then a long strand of DNA will be twisted up, and so sometimes it's called a twisted ladder. We say DNA gives us our traits. So how do we get from a strand of DNA all the way to an actual trait, like the color of our eyes? Well, it all depends on proteins. And so protein synthesis is the process of how the message of DNA is transcribed and then translated into protein. So synthesis just means making. So if you're trying to remember what protein synthesis is, just think about how we get proteins, how we make proteins from DNA. Let's see how this works. So remember, the DNA is in the nucleus of eukaryotic organisms. So we have our DNA strand, it's going to be split apart, and it's going to start to be copied as mRNA. And so this mRNA strand will build, basing itself off of part of that DNA strand. There's a couple of differences between mRNA and DNA, and one of those things is that instead of T's, RNA has U's. So when we pair with A, we're going to match it up with a U instead of a T in mRNA. RNA is also single-stranded, so once it's done copying the DNA, it'll leave the nucleus, the DNA will go back together, and then the mRNA will reach the ribosome where we can do the rest of the work, the translation part, where we're going to get our tRNAs, which are another type of RNA, are going to match up with the message on the mRNA and bring over these little molecules called amino acids. So glue here, that's one amino acid. And as these tRNAs match up with the code on the mRNA, we're going to bring more and more amino acids over, and they will be linked up by bonds here. These are peptide bonds, and we will start to form a protein. And once we have our amino acids linked together, we have our polypeptide or our many peptide chain, and that will result in our protein, which will do the job for the cell that we need it to do. It gets very long and it folds up in a specific shape, so it becomes a functional protein, like an enzyme, for example, or a pigment protein that'll color our eyes. So again, some differences between DNA and RNA. DNA is double-stranded. Think of that D for double. RNA is single-stranded. DNA has actually a different sugar than RNA. It's a deoxyribose sugar. RNA has a ribose sugar. And then, of course, DNA has T's, where RNA has a U as one of its bases. So let's practice here. If you want to pause the video and write down the mRNA sequence you think would go with this DNA strand, go ahead. I'll give you a second. And here it is. This is the RNA sequence that would come from this DNA strand. So now we have a message in our mRNA that can be taken out into translation. Now, if you confuse transcription and translation, remember transcription, we're changing from one type of nucleic acid, a deoxyribonucleic acid, into another type of nucleic acid, a ribonucleic acid. We're transcribing it into a message. So thinking about you writing down a message that you're going to pass to your friend, that's transcription. Translation, on the other hand, if you're translating something from English to French, you're changing languages, and that's what we're doing here in the cell as well. We're changing molecular language, and so we're going from a nucleic acid to an amino acid chain or a protein. So if you want to remember which one's transcription and translation, think about which one's writing down the message, that's DNA to mRNA, and which one is getting us our chain of amino acids, or we're changing our language. We're going from a nucleic acid to a protein, so that's translation. Now, for the SOL, you might be asked to read something like this called a codon chart or an amino acid table, where we're given all these letters here and it tells us which amino acids go with which 3 groups or codons of RNA. So for example, if our codon was UCA, we would start here in the center with the big letter U, and then we'd find the C in the next level, and then we would go to the outermost layer, A. Okay, so that's serine, or SER. This is an abbreviation for an amino acid. Let's try another one. How about AAA? So we start here in the center, A, and then we look here to see which one this touches. A, that's lysine. That's another amino acid. So every codon matches with a specific amino acid. Now there's other types of codon charts as well. You may see one that looks like this. You have to read them a little bit differently, but sometimes they're broken down very helpfully. Okay, so now we're getting to cell structure and function, where we're going to talk about the cell theory, some organelles, the cell cycle, cell transport, and cell specialization. Here we go. So for the cell theory, we know that all living things are made of cells. Cells are the basic units of all living things, and all cells come from other cells. All cells also are surrounded by a cell membrane, which is a phospholipid bilayer. It's made out of lipids, one of those main categories of organic compounds. All cells have genetic information, so DNA, we talked about that, and all cells have cytoplasm, or something to hold its materials in. Now we get more cells. All cells come from other cells through a process called mitosis, where the cell divides and splits its genetic information into 2 new identical daughter cells. If you need to remember the steps of mitosis, PMAT is a helpful acronym for prophase, where the cell is preparing and getting ready for cell division; metaphase, where the duplicated chromosomes line up in the middle of the cell; anaphase, where the sister chromatids start to be pulled away from each other; and telophase, where a new nuclear membrane forms. We have our genetic material entirely separated; and then cytokinesis, which is not in the PMAT acronym, is where the cytoplasm actually cleaves or splits down the middle and we get 2 new cells. So prophase, metaphase, anaphase, telophase, and cytokinesis. Now remember that this is just part of the cell cycle, or the steps a cell goes through in its normal life cycle. For human cells, this takes place about once every 24 hours, but it can be different depending on the type of cell. Cells spend most of their lives in interphase, or the period between mitosis, between cell division. So we have G1, S phase here. S phase is when the cell duplicates its DNA actually, and then G2. So getting into cell transport, we need to talk about how organisms maintain homeostasis at the cellular level, and a lot of this involves which molecules can get in and out of the cell. One way is through diffusion, and I also have a cell transport review video, so you can go back and check that out. Diffusion is just the movement of molecules from a high to a low concentration. So when we have a higher concentration here, they're going to naturally move in the direction where there is a lower concentration until an equilibrium is reached, which we call an isotonic solution, if there is equal amounts of solute or particles on either side of a membrane. All right. There's also facilitated diffusion, where molecules are going to diffuse high to low still, but with the help of a transport protein. And then there's active transport, where we need to go in the opposite direction of the concentration gradient. So we're going from low concentration here to high, which again is not the way they're naturally going to move. So ATP, or cellular energy, needs to be used. And so this is done also with the protein. We're going from low to high, and we need to use energy. So another type of cell transport, which is a type of diffusion, is osmosis. And this is specifically the movement of water from a high concentration to a low concentration. H2O for water, O for osmosis. A hint to get this right is to think about the phrase solute sucks. So let's talk about what that means. So solute are any of these particles that are dissolved within the solution. In most cases, when you get into these water transport problems, it's going to be water. And so these pink dots are our solute. Right here we have what's called kind of an equal concentration of solute particles inside the cell and outside the cell. So we call this iso for equal, or isotonic. Now, water is going to continuously move across the membrane here back and forth, but it's going to be pretty equal movement, so nothing much is going to happen to the cell. But now we have a change in the number of particles both inside and outside the cell. So let's think about where there's more particles first. So we have a higher concentration of particles inside the cell, a lower concentration outside. But water, it's the reverse. So there's actually a lower concentration of water here than there is here outside the cell. So think water is higher here and lower here. So water will move from high to low, but that's a lot of backwards thinking. So an easier way to skip a step, if you think about where there's more solute, water is going to go in the direction where there's more solute, because solute sucks. So water will go into the cell here. This is also called a hypotonic environment. Think because there's a low solute concentration on the outside. So hypo kind of rhymes with low. And so think that water is going to go from the hypotonic environment into the cell because solute sucks. All right, here's the opposite. We have more particles outside the cell than we do inside the cell, so it's a higher concentration out here. It's hyper, it's exciting, it's like a party, so it's called a hypertonic environment. Now remember, if solute sucks, where's the water going to go? Water's going to go outside the cell. So let's do a problem together. A cell is in a beaker with a solution with a higher salt concentration on the outside than on the inside. What is going to happen to the cell? Think about it. Well, think about where there's more water. There's a higher concentration of water inside the cell. Water's going to leave the cell probably, and over time, if a lot of water leaves, that'll cause the cell to shrink. In the opposite direction, if water moves into the cell because we're in a hypotonic environment, then the cell could expand or even burst. Now, a main theme in biology is that structure dictates function or form fits function. So think about all the ways that molecules and cell parts and body shapes are in a specific form for a specific job. For example, all the folds in the brain, the gyri and the sulci, allow for increased surface area to have lots of connections between neurons and be able to send messages and think lots and lots. The same thing goes with the increased surface area and all the folds in the intestines. The more surface area, the more opportunities we have for absorption and particle exchange. Now, we know that in our bodies we have lots of different types of cells, but during development our cells become specialized and they actually turn into different shapes. So our cells don't actually look like this typical cell diagram drawing. They're going to look very different, from things like our neurons to our red blood cells. And think about how form fits function. So our neurons have lots of little branched dendrites here, and these are going to be able to receive signals from other neurons that they're going to send in across these synapses, and they're very long so they can propagate messages down these tails. So think about form fits function, or how structure dictates function, as you go through your SOL review. Let's talk a little bit more about what's inside those cells. Let's do a quick jump to our organelle review. Organelles are structures within cells that have specialized functions. And you probably remember a lot of organelles from previous science classes, but we're going to focus today on some of the main ones you'll need to be able to know and recognize by picture, function, or name. So let's look at these two pictures. We have the nucleus, drawn simply here, plasma membrane, which is the surrounding double layer around the cell, which if we zoomed in closely, we see is a phospholipid bilayer. The cell wall with plant cells and certain prokaryotic cells, and mitochondria, this oval with the squiggly line, our vacuoles here and here, chloroplasts, again only in plant cells, and ribosomes, represented frequently by little dots. Sometimes you'll see a mitochondrion drawn very technically, and they look a little bit more like this than this simplified drawing here, but sometimes you'll just see an oval with a squiggly line. So you need to be able to recognize both types of pictures. And you'll also potentially see a chloroplast drawn more like this than the one I have in my picture. These are stacks of thylakoids, which are an important part of the chloroplast. Just be able to recognize chloroplast as an important organelle with these kind of stack-like structures within a plant cell only. So getting back into the differences between animal and plant cells, which you might remember again from other science classes, plant cells have cell walls, and they look kind of like these geometric shapes. Animal cells do not. Plant cells also have chloroplasts, where animal cells do not, and plant cells tend to have one large vacuole for water storage, where animals can have smaller vacuoles or multiple vacuoles. So make sure you recognize those main differences between plant and animal cells. Those aren't the only differences, but those are key important ones. So recognizing our organelles by picture, again, here's our simplified mitochondrion. This is for energy, so you need to be able to understand that the mitochondria is where the cellular energy is created or made in a process called cellular respiration. You need to know more than just the mitochondria is the powerhouse of the cell. Here are simple circles with kind of a blank space in them. That would be a vacuole, which again is for storage. Our ribosomes are represented by little dots, and this is where proteins are made. Our nucleus is going to be in the center of the cell. Sometimes you'll see chromosomes if the DNA is condensed represented in them, but it's going to store our DNA or our genetic information in eukaryotic organisms, and we'll get to that in a second. Our cell membrane is going to be represented, if we zoom in closely, by this phospholipid bilayer, and its purpose is to provide a semi-permeable barrier, letting some things in and some things out of the cell in order for the cell to maintain homeostasis and do everything that it needs to do. Our cell wall is going to provide structure and support, another layer of protection of the cell, again not in animal cells, and it's going to be kind of geometric in shape. And our chloroplast is the site of photosynthesis, only in plant cells, so we're not going to see these in animal cells. Now if we look back at our
plant and animal cells, these are both eukaryotic organisms, meaning they have a true nucleus, or they have a nucleus and other membrane-bound organelles. So organelles like the mitochondria, organelles like vacuoles, and these are generally larger and more complex. Prokaryotic organisms are more primitive. We think they evolved first. They typically only have a few key features. So they're very simple, they're very, very small, they're more abundant on Earth than eukaryotic cells, but they're very different. So in prokaryotic cells, we do not have any of those membrane-bound organelles. We don't have a mitochondria, we don't have vacuoles, we don't have a nucleus. Instead, the DNA is just free-floating within the cell in a feature we call the nucleoid. We do have ribosomes because all cells do make proteins and have to do protein synthesis. We do have a cell membrane to contain the cytoplasm and the essential functions of that and the essential features of that cell. And that's about it. Sometimes there'll be external features like our flagella, which is for movement, or cilia, also for movement. Those are small hair-like structures. I'll show you a picture of one of those later. And those can exist in prokaryotic organisms, but again, not all prokaryotic cells have those. So again, prokaryotic organisms, much more simple, no nucleus, no membrane-bound organelles, very, very simple features. Plant and animal cells are both eukaryotic.
That was a really quick review of some of the really main important ideas for understanding your information for your biology SOL. Thanks so much for watching. If you want to get more SOL review, make sure to subscribe and stay tuned for more videos in the series. We're going to finish up all of the biology content for the Virginia SOL. Thanks so much for watching. Give this video a like if it's been helpful, and I'll see you later.